Assalamu alaikum. Hello, my dear students. How are you all? Welcome to another biology class. I'm your biology teacher, MD Jahidul Hassan. Today, we're going to learn from the chapter Human Immunity. And today, we're going to talk about the topics antigen, antibody, and its structure, vaccine, type of vaccines. So, let's start our class. So, what is antibody? Antibody is any of a large number of proteins of high molecular weight that are produced normally by specialized B cells after stimulating by an antigen and act specifically against the antigen in an immune response. Okay, and the antibody producing cells are the B cells or B lymphocyte cells. I've already discussed about the B cells and B lymphocytes. I know in last lectures. Okay, so here the Paul Ehrlich coined the term antibody uh, mm, in 1897. Actually, antibodies are large chemical molecules of glycoprotein. Do you know what is glycoprotein? Glyco means carbohydrate, proteins means protein. So the glycoprotein means the complex structure, which is a combination of carbohydrate and protein. And this glycoprotein, that means the antibody, which is secreted from the B lymphocytes or plasma B cells or plasma cells of the blood. Okay, and this antibody is also called the immunoglobulins, and the short form is Ig. Okay, immunoglobulins. And if necessary, each B lymphocyte of our of the blood cell can produce thousands of antibodies per second. So the rate of antibody production is very rapid, very high. So the human body able to produce almost about 100 million types of antibody, and each type of antibody is unique, and it can defend the body against one specific type of antigen. Okay, so here the immune antibody, immunoglobulin or Ig or the gamma globulin is the part of the blood. Okay, so now always these two terms comes together, antibody, antigen. So what is antigen then? Okay, antigen is a substance says which recognized as a foreign substance, okay, when introduced into the body, and this thing is capable of stimulating an immune response. Okay, so antigen is also known as immunogen because it can stimulate an immune response. Okay, and antigen includes all the parts of bacteria, viruses, and other microorganisms like the coats, the capsules, cell walls, flagella, fimbriae, and toxins. All these things are also included in the antigen. And this term was first actually coined by the Ladislas, a scientist, in 1903. Okay. Lipids and nucleic acids, it can also be the antigen. It can also show some antigenic characteristics. Uh, mm, and even there are some antigens which is the combination of lipids and proteins and nucleic acids and proteins and polysaccharides. So non-microbial, non-self antigens can include the pollen, egg white, and proteins from transplanted tissues and organs, or on the surface of the transfused blood cells. That means actually any kind of foreign particles which is not the self of organisms of an organism body, you can call them as antigen. Okay, so there are two types of antigens, one is exogenous antigen, another one is endogenous antigen. Here the exogenous antigens are antigens that have entered the body from the outside, for example by inhalation, by ingestion, or by injection. Okay, such as bacteria, viruses, fungi, protozoa, helminthes, and foodstuffs, pollens, all these things are the exogenous antigens. And what is the endogenous antigens then? Endogenous antigens are generated within normal cells as a result of normal cell metabolism. That means the endogenous antigens are produced within the body, within the cell of an organism. Okay, so and these endogenous antigens can be produced due to the viral or bacterial infections. And but the thing is, these endogenous antigens produced within the cell as a result of normal cell metabolism. Okay, so mm, some examples like tumor specific antigens, protein antigens, histocompatibility complex antigen. Okay, so many types. Okay, now let's talk about the structure of an antibody. The structure of an antibody is complex, and this is our, see here, this is the Y-shaped structure. This is an antibody. Uh, and here, this one is actually the immunoglobulin or antibody monomer, and usually it is Y-shaped molecule, and that consists of four polypeptide chain. See, one, two, three, and four. These four polypeptide chains, all these things actually connected by disulfide bond. See here, this chain is connected with this chain by this disulfide bond. Do you mean di? You know, di means two, sulfide means sulfur. So disulfide bonds here, this the disulfide bonds, and this chain is also connected with this chain through this disulfide bonds, and this one is also connected here with this disulfide bond with with this chain. So that means all these polypeptide chains are connected by each connected to each other by these disulfide chains. So disulfide bonds actually. Okay. So each chain is composed of structural domains called immunoglobulin domains. Each chain is called immunoglobulin domains. Here the heavy chain with see, this one is the heavy chain. Why they are heavy? Because they contains more amino acid than this chain. This light chain, these two chains are called the light chain because they are smaller, because they have less amounts of amino acids than these large chains. Usually, these light chains it contains around 200 amino acids, but these heavy chains they contains around 440 amino acids. Okay, that's why they are more heavy, that's why they are called heavy chain, and they are more light than this heavy chain, that's why they are called a small chain or light chain. Okay, and there are two types of region here, both of this light chain and heavy chain. Okay, and this region are called the variable region and constant region. Okay, these regions, this dark deep blue, these deep blue regions are the variable region, and this light blue, this means, that means this here these two regions are the variable regions. These two regions are the variable regions, this deep blue region and this light blue regions. These light blue regions are constant region. Okay, variable region means this the change the shape of these areas are changeable. Okay, but the shape of these areas are constant, no change can take place here. Okay. Mhm. So that's why these regions are called. So the variable region of the light chain is called the VL region, and the variable region of heavy chain is called the VH region. So that means this region and this region are the VH region, and this region and this region are the VL region. And similarly, the constant region of the heavy chain is called CH. So that means these two region here, these two regions are the CH region, and this region of the light constant region is called CL. Okay. So and here and the thing is, these portions of the heavy chain is called the hinge region. Okay. Because here these portions are the flexible portions. Okay. This heavy chain can move here, these two chains can move here in these portions, in this position, this way. These are called the hinge region. Okay. So mm, now say say this region, that means the portion, this portion of the antibody, and this portion of the antibody, this region is also called the Fab region. So what is Fab region then? Fab region means fragment antigen binding region. Fragment antigen binding region or Fab region, because each of the two expanded shape of the Y rod shape Y structure of the antibody, this region actually it contains both the variable and constant region the polypeptide chain, and actually this region actually this region is only interact with the antigen. That's why this region is called antigen binding region. So this total these portions are called fab region, and here these portions, that means the heavy chain portions and the lower portions of the heavy chain and also the constant regions, that is from the disulfide bond that's below, that's beneath these disulfide bonds, these regions are called the FC region, fragment crystallizable region, or FC region. Okay, okay. Now see, and see here, this is here is an antigen, here is another antigen. A single antibody can interact with two antigens simultaneously, and the region where this antigen antibody interaction occurs, this region is called like all, or can the site, or this site, this portion of the antibody, this portion is also known as paratop. Okay, this notion is also known as paratop. That means the portion of the antibody which can interact with the antigen is called the paratop. Due to the recombination of amino acids in this paratop region, here the random arrangement or variable variation can take place. Okay, that's why this region is also called the VDJ, variable diversity joining region. Okay, why? Because this portion of the antibody can rearrange themselves according to the shape of the antigen. Okay, so this is the overview of the this is the overview and the overall structure of an antibody. So now let's talk about the types of antibody. In human blood serum, there are 5 types of antibody or immunoglobulin molecules. And these types of antibody generally determined by sequence of amino acids in heavy chain of antibody. There are 5 types of heavy chains in human antibody based on sequence of amino acids, and they are, see, they are called the gamma chain, mu chain, alpha chain, epsilon chain, and delta chain. According to these chains, we can call them the we can call the gamma chain containing immune and antibody as immunoglobulin G, and also the mu chain containing immunoglobulin is called the immunoglobulin M. Immunoglobulin M. Okay? And immunoglobulin A, it contains the alpha chain, and immunoglobulin E, it contains the epsilon chain. Okay, and the delta chain is contained by here the immunoglobulin D. So let's talk about the immunoglobulin G. Immunoglobulin G, almost 75% of the total immunoglobulin or antibody of the blood in the blood is actually this type of antibody, immunoglobulin G. This is the most abundant type of antibody which is present in the blood. It's kind of antibody that works efficiently to coat microbes, speeding their uptake by other cells in the immune system. And this immunoglobulin, this antibody, it protects the body against gastrointestinal and respiratory problems. It is commonly found in the milk and also in the saliva. Okay, now the immunoglobulin E. This is a very rare type of antibody, immunoglobulin E. On less than 0.1% of all antibodies which is present in the blood is the immunoglobulin E. It is involved in allergic and hypersensitivity reactions. Okay, and it also provides protection against parasitic worms. And now the immunoglobulin D. This is also another rare type of antibody, and this type of antibody is found in blood, lymph, and B cells, and plays a very important key role in initiating the early B cell responses. Okay, and these two, immunoglobulin A and immunoglobulin A. Immunoglobulin M, almost 5 to 10% of all the antibodies in the blood are immunoglobulin M. It is the largest antibody in the blood which is present and which is produced, secreted by the plasma cells. It is also known as the macroglobulin because it consists of 5 times. Actually, see here, it is the 5 times larger than this immunoglobulin G. 1, 2, 3, 4, 5. So that means actually 5 immunoglobulin G and antibodies, when they compact with each other by disulfide bonds and also joining chain, present within them, they can form this immunoglobulin M. And the another type was immunoglobulin A. Almost 10 to 15% antibodies of the serums are immunoglobulin A. Uh, and this type of immunoglobulin A, it also second most abundant abundant type of immune antibody. It levels it decrease the levels of immunoglobulin A decrease during stress, lowering resistance to infection. Okay. Immunoglobulin A found in the colostrum. Colostrum means the first meal which is secreted by a mother with huge amount. Okay. Okay. Now antigen antibody reactions. How this antigen and antibody they react with each other, and how those antibodies actually neutralize the antigens or destroy antigens. Mm, uh, so antigen antibody reactions are different types, and one type of antigen antibody reactions are agglutination. Okay, they are agglue. See the term glue. Glue means joining. So aggregation of microbes, aggregation of microbes or cells due to antibody binding is known as the agglutination. Immunoglobulin M is more responsible for this type of agglutination. The antibodies that cause agglutination of cells are called agglutinins, and the antigens aggregated are called agglutinates. Okay. So now the opsonization. Opsonization means neutralization. So the antibody that is making the antigen susceptible for phagocytosis is known as opsonin. Okay. Remember the term opsonin. And now certain antigens escape the phagocytic reaction through various ways. If any antibody binds to such antigen, it becomes susceptible for phagocytosis. This process is known as opsonization. Okay. Therefore, anything coated with these antibodies will be bound by macrophages and engulfed. Okay. Um, this is called the opsonization process. And another type of antigen antibody reactions is the activation of complement system. Okay. Antibody binding with antigen is not sufficient to remove the antigen from body. Okay. Antigen and antibody complex initiates the activation and binding of complement system to it. Binding of complement system to antigen antibody complex is known as the complement fixation. And the activated complement enzymes then continue further immune reaction that finally removes those foreign antigen, foreign agent. Okay, this is the process. And another type of antigen antibody reaction is the lysis. The complement activation can result in lysis of animal cells and gram-negative organisms. Thus, complement fixing antibodies, immunoglobulin M, may cause this type of lysis of susceptible targets. Okay, lysis means the destruction, breakdown. And the final type of antigen antibody reaction is the neutralization. Neutralization is the immunological sense, which refers to the ability of antibodies to block the sites on bacteria or viruses that they use to enter the target cells. So, thus, some of the antibodies can block viral infection by neutralization, which is usually the result of a formation of virus antibody complex. Okay. So now you can ask me, okay, these are the types of antigen antibody reactions, but how they interact with each other. Okay, actually, in our last slide, like see here, these are the paratop region where these antigen antibody interactions occurs. Okay, so these paratop regions, these variable regions actually change its shapes according to the shape of the antigen. Thus, this antigen and antibody can act with other, can interact with each other, just like the mechanism of lock and key mechanism. Lock and key, how they interact with each other. They have similar types of. They have just the opposite types of shapes. Okay, so so, so same thing happens here in the interaction within the interaction of antigen antibody during the interaction of antigen and antibody. Okay, so now let's talk about the vaccine. Very important, and nowadays we are very aware of it. Vaccine due to the corona situation. Vaccine is a preparation of killed microorganisms, living attenuated microorganisms or living fully virulent organisms that is administered to produce or artificially increase immunity to a particular disease. So, a very interesting things. Vaccine actually, vaccine typically contains an agent that resembles a disease-causing microorganisms and is often made from weakened or killed forms of the microbes, or its toxins, or its surface proteins, or its any kinds of parts. Okay, so thus introducing these portions or these killed or weakened microorganisms, we actually we are able to actually increase the immune response, or we are able to create a memory cells. Thus we can protect ourselves from those foreign invaders, okay, for further infections, for further attack, further infections. Or actually inoculation of a vaccine into body to prevent a disease means that inoculation of microbes responsible for that particular disease. Okay, so now let's talk about some properties of an ideal vaccine. Actually, it gives lifelong immunity by producing memory cells, and it's also broadly protective against all variants of organisms. It prevents disease transmission from one person to another person. It also rapidly induces the immunity. It transmits maternal protection to the fetus. Okay, and not need to be administered by injection. In some cases vaccine is, it's not necessary not to be administered by injection. Okay, um, some vaccines are nowadays can be introduced into the body by adhering a patch on the skin even. So the another property is stable, cheap, and safe. Okay, and the first vaccine was discovered by Dr. Edward Jenner in 1796 against smallpox. Now, what are the principles of vaccination? Vaccination is the administration of antigenic material or vaccine to stimulate an individual's immune system to develop adaptive immunity to a pathogen. So these are the principles. Now, the first thing is vaccine consists of attenuated or killed microbes or their byproducts. Okay, and then attenuated microbes, when used as vaccine, they are able to multiply within body after inoculation, but they are not able to produce any diseases. So attenuated toxoid vaccine could not produce any damage of the body. The first, then the four most important principle of vaccination are the introducing vaccine um, must not be able to produce any kinds of disease. Okay, so the third principle of vaccination are immunity developed from vaccination do not last lifelong, but its lifespan may extended by booster dose. In some cases, this is the scenario of some vaccine, not all the vaccines. There are some vaccines which can induce a lifelong immunity, and there are some vaccines which can um, which to induce lifelong immunity we have to inject, or we have to uh, use another more booster doses to sustain long-term immunity. Okay, and the durability of immunity sometimes depends on the ways of vaccine administration. For instance, vaccination gives instant permanent result when applied through the alimentary canal in case intestinal infection. Okay, and the another principle of vaccination is the immune response period is much longer than the incubation period of microbes. Hence, vaccination is not effective after infection of disease, with one exception of hydrophobia. Like because incubation period of hydrophobia is very long, Okay, so now let's talk about types of vaccine here. There are different types of vaccines like killed vaccine. Killed vaccine means these are previously virulent microorganisms, but then have been killed with chemicals or heat and then introduced within the body, such as influenza vaccine, a type of killed vaccine. Attenuated live vaccine. Attenuated live vaccine is the type of vaccine that live microorganisms are used here. and they're cultivated under specific conditions, but they're disabled. That their virulent properties have been disabled. And they typically provoke more durable immunological response, and they're preferred for healthy adults. Okay, such as measles, mumps, rubella, influenza vaccines are the attenuated live vaccines. Toxoid vaccines. That means the toxins separated from bacteria are inactivated to make as vaccine. This type of vaccines are called toxoid vaccines, and this technique is reserved for disease in which the secreted toxins is the main cause of the illness. Okay, so then that means when we use the toxic substances from the virus or bacteria to induce our immunity, is called the toxoid vaccines. And the subunit vaccine, subunit means the portions of the surface proteins or the body proteins of our microorganisms. When we use those subunit of a vaccine to induce our immunity, we can call those vaccines as the subunit vaccines. Okay, hepatitis B vaccines are the example of subunit vaccine. Conjugated vaccines are made using a combination of two different components. Okay, conjugate vaccines, however, are made using pieces from the course of bacteria. Actually, conjugate means conjugation. That means the combination of two things. When you combine two things, like a portion from bacteria or virus and another portion from um, uh, it can be protein, it can be carbohydrate, it can be lipid, whatever. So surface chemical vaccine. Oh, so example of conjugated vaccine is, see here, the Haemophilus influenza vaccine. And surface chemical vaccine, in some cases we can use the surface proteins of the viruses to induce immune response. This type of vaccine is called surface chemical vaccine. Hepatitis B vaccine can be the example of surface chemical vaccine. And the vaccine, DNA vaccine. Even we can use the germ's DNA to induce our immunity. Okay, scientists they hope to extract a portion of the germ's DNA and make copies of it to create a DNA vaccine. And this type of vaccine would help hopefully produce better protection like a live virus vaccine. But with even less likelihood of producing diseases. These are currently studies of DNA vaccine for herpes and influenza. Okay, mm, I think that's all for today. Mm, Thank you all.